The overall goal of the following experiment is to develop new fluorescent sensors based on dendrimers, with improved properties for pH imaging in vitro in living cells and in vivo. This is achieved by first conjugating pH-sensitive dyes to a dendritic scaffold together with other moieties, such as targeting groups or pH-insensitive dyes, and in vitro measurements are taken to generate a pH calibration curve. Next, to image pH in living cells, the sensor is delivered by electroporation, and pH maps are generated using confocal microscopy. To image pH in the extracellular space of the brain during physiological and pathological neural activity, the sensor is microinjected into the brain of anesthetized mice. Fluorescence data is then acquired using two-photon microscopy. Results obtained show that Dendrimer-based sensors succeed to measure pH changes with high spatial and temporal resolution, both in living cells and in vivo.
Although several fluorescent dyes with pH-sensitive behavior have already been employed, they suffer with several drawbacks, such as poor solubility and cell leakage. Moreover, they do not allow the ratiometric absolute measurement. Here, we demonstrate how the conjugation of pH-sensitive dyes to a dendritic scaffold enhances the performances, allowing accurate measurement with a spatial and temporal resolution. This method could be a useful tool to study physiological and pathological processes regulated by pH in living cells.
The disruption of the pH regulation in the neural system is associated with several conditions, like strokes and epilepsy. An available pH sensor proved to be poorly effective, leading to the design of the presented sensor.